So who is the most famous scientist on campus? You probably will say Mario Capecchi, who won the Nobel Prize here in Utah. Mario Capecchi designed an experiment to make a mutant by removing one gene from one animal. And by analyzing the difference between the mutant and the normal animal, we can understand the function of that gene. In our lab, we decided to make a zebrafish mutant called LEF1, and by removing LEF1 from zebrafish. And I will tell you why. Zebrafish is a great animal model because it's a vertebrate, so it shares many genes with human. And also, its advantage is one breeding of one parent zebrafish will give rise to many hundreds of offspring for experiment. So our lab is interested in wing signaling pathway because it's important in development and also in disease state. We know that wing signaling pathway activates LEF1, and the LEF1 activates many other genes, but we don't know what are these genes and what are the function of these genes. So the goal of my thesis is trying to ask this question by studying LEF1 mutant in zebrafish. So we made the mutant, and I find that the zebrafish mutant has a smaller hypothalamus, which indicates that LEF1 is required for normal formation of hypothalamus. Hypothalamus is an important brain structure in the brain and has many functions such as feeding, reproduction, and body temperature control. And to ask what genes are activated by LEF1, I dissected the hypothalamus from hundreds of embryos and sequenced the whole genome. And I find that many genes are no longer present in LEF1 mutant, indicating that these genes are actually activated by LEF1 in zebrafish. And the next goal is trying to make mutants for this gene and study their function. And I believe that the significance of my work is trying to help identify new rules and roles of the hypothalamus, and this will also be clinical relevant. But people will start to ask, is what I find in zebrafish also present in human? To ask this question, I decided to make a mouse mutant. Mouse is also a mammal and more evolutionarily closer to human. So preliminary studies show that the mouse hypothalamus is also reduced in the mutant, indicating that this function is evolutionarily conserved in mouse, probably as well as in human as well. And one preliminary study I did this month suggests that the mouse mutant is resistant to anesthesia. And this would be the first evidence that hypothalamus function as the mechanism of anesthesia. And this will help design better anesthetics for surgery. So I believe that this example will convince you that my thesis work will be significant for all the people here. Thank you.